competitive inhibitor of the B7 CD28 co-stimulation, a T lymphocyte associated protein called cytotoxic T lymphocyte associated antigen 4 or CTLA4 also has a high affinity for B7, which allows for self-tolerance or non-responsiveness to self-antigens. Its clinical application in rheumatology and transplant rejection allows CTLA4 immunoglobulins to fuse with the Fc fragment of human immunoglobulins and bind and block B7 molecules to inhibit the immune response. B cells are genetically programmed to respond to only one antigen and also become activated in two steps. The coupling of the B cell receptor and the antigen signals the cascade of events leading to the presentation of antigen peptides on the MHC2 molecules to be recognized by the complementary T cell receptor on a helper T cell. The second signal is when CD4 molecules on B cells interact with the corresponding ligands on the helper T cells, resulting in the release of cytokines that lead to the differentiation of B cells into either plasma or memory B cells. Plasma cells synthesize antibodies, while memory B cells are responsible for subsequent reinfections with the same antigen. Natural killer cells are activated by gamma interferon and equipped with Fc receptors that recognize and destroy corresponding antibody-coated cells. This identification and resulting apoptosis can also be mediated by the complement system. Components of this system, usually C3, binds to bacteria and foreign cells and act as ligands for macrophages to destroy the pathogens. After antigen binding, CD8 T cells divide and differentiate into effector cells. These cells contain secretory vesicles rich in fragmentins. Perforins, for example, create transmembrane channels on target cells that allow proteases, such as granzymes, access into the cells that then activate caspases, effectively leading the cell into apoptosis. In order to ensure that the immune system is controlled and to prevent autoimmune diseases, mechanisms are in place to limit its response. Activated T cells possess CTLA4 receptors that inhibit the co-stimulatory signal, and regulatory T lymphocytes produce specific cytokines that suppress both T and B effector cells. These play a role in the delayed hypersensitivity reactions, such as in MAN2 testing in TB, and to ensure the survival of organ and tissue grafts. The different antigen-presenting cells perform their function of facilitating antigen interaction with helper T cells by initially endocytosing the pathogens and breaking them down into smaller peptides. These peptides are coupled with their corresponding MHC molecules and instated on the plasma membrane of antigen-presenting cells to interact with the CD4 cells. Classically activated macrophages are those activated by both the action of gamma interferon and co-stimulated by the interaction of CD4 T ligand and CD4 on Th1 helper T cells. They lyse antigens and promote inflammation and tissue destruction and apoptosis. They are also capable of sequestering immunologically inactive organic and inorganic particles like carbon, pigment, and TB bacilli, fusing together to form multinucleated Langerhans or giant cells. In contrast, alternatively activated macrophages are those that are acted upon by interleukins produced by Th2 lymphocytes and have the opposite effect of anti-inflammatory properties, helping instead to rebuild tissues. We now move on to the lymphatic vessels. Lymphatic vessels drain fluid and larger molecules from the extracellular spaces back into the circulation. Lymphocytes in the lymph enter the lymph nodes through afferent vessels, while those in the blood enter through postcapillary venules, or what we call the HEVs, high endothelial venules. In the lymph nodes, specialized dendritic cells trap antigens to be processed by antigen-presenting cells. Activated lymphocytes then leave via the efferent lymphatic vessels, leading to the right lymphatic trunk or thoracic duct, both emptying into the junction of the internal jugular and subclavian vein. Diffuse lymphatic tissues are accumulations of lymphatic tissues not enclosed by a capsule and are located in the lamina propria, strategically positioned for initial contact with antigens. Because of their location within the mucosal linings of tracts, they're often called MALT or mucosa-associated lymphatic tissue. These tissues protect the body through local antibody secretion and are responsible for chronic inflammation and hypersensitivity reactions. The photo shows MALT in the lamina propria of large intestines with a predominance of lymphocytes, as well as some fibroblasts, plasma cells, and eosinophils. Similarly, lymphatic nodules or follicles are defined but unencapsulated, localized concentrations of lymphocytes found in the walls of tracts. They can be primary or secondary, depending on the formation of zones. Secondary nodules contain germinal centers composed of proliferating B lymphocytes differentiating into plasmablasts, which appear lighter due to their dispersed euchromatin. The formation of this lightly stained germinal center indicates tissue response to antigen, called the germinal center reaction. Next to this zone is the corona or the mantle zone, appearing darker because of the small lymphocytes not previously stimulated by antigens. Lymphatic tissues and nodules along the alimentary canal are collectively called gut-associated lymphatic tissue and include the lingual, pharyngeal, and palatine tonsils, the Peyer's patches in the ileum, and the vermiform appendix at the base of the cecum. When located in the respiratory system, these lymphoid organs are termed bronchus-associated lymphatic tissues and mucosa-associated lymphatic tissues and many other mucosal surfaces exposed to the internal environment capable of forming an immune reaction to antigen exposure. Lymph nodes are encapsulated lymphatic organs located along the path of the lymphatic vessels and serve to filter lymph and process antigens to activate lymphocytes into effector cells. Lymph enters the nodes through afferent vessels and leave via the efferent vessels through the hilum. These nodes are surrounded by a dense connective tissue which extend as trabeculae into the cortex. The rest of the organ is composed of reticular cells and fibers that support it. The lymph nodes are divided into the outer cortex and inner medulla. The superficial nodular cortex contains primary and secondary nodules with germinal centers, containing mostly B cells, and the deep cortex under it contains most of the T cells and is also called the thymus-dependent cortex. The medulla contains lymphatic cords that house the cells of the lymph node and medullary sinuses that converge and drain at the hilum into the efferent lymphatic vessels. A few types of cells that contribute to the immune response have been identified in the reticular meshwork. The reticular cells are fibroblasts that secrete type 3 collagen or reticular fibers and ground substance. They wrap around bundles of reticular fibers with their processes, express surface molecules, and produce chemokines that attract lymphocytes and dendritic cells. Dendritic cells monitor the local environment and are highly efficient antigen-presenting cells containing MHC molecules that recognize, process, and present antigens to T cells, and as such are found in T lymphocyte-rich areas. Macrophages are primarily phagocytic, capable of endocytosis and digestion, and because they contain MHCs, can also perform as less efficient antigen-presenting cells. Follicular dendritic cells have processes that interdigitate between B lymphocytes. They retain antigen on their surfaces, but are neither capable of endocytosis or of antigen presentation because they lack MHCs. This is how lymph circulates in a lymph node. Afferent lymphatic vessels drain into the subcapsular cortical sinuses that continue down into the parenchyma as trabecular sinuses, which then in turn drain into deeper medullary sinuses. These sinuses are lined by endothelium, where they face the capsule and trabeculae, but are discontinuous facing the parenchyma. Through these discontinuations, macrophages send their pseudopods for monitoring. Apart from these processes, reticular cells and fibers also crisscross the sinus lumen to enhance filtration and act as a mechanical filter. Majority of lymphocytes, however, enter the nodes through the HEVs or the postcapillary venules in the deep cortex. Their endothelial cells express adhesion molecules that interact with L-selectin and integrins on lymphocytes, and chemokines CCL, chemokine motif ligand 19, and CCL21 that attract T lymphocytes into the lymphatic tissues, and also CXCL13 that attract B lymphocytes. These chemokines play a role in the regional distribution of lymphocytes in the lymph node parenchyma. HEVs also contain water channels with aquaporin-1 molecules that cause rapid resorption of interstitial fluid. The immune response is initiated by the phagocytosis of foreign material by macrophages and antigen-presenting cells, which then lead to the activation of lymphocytes. Activated B cells differentiate into plasma cells, memory cells, and plasmablasts, which are capable of antibody secretion. Plasma cells migrate into the medullary cords, while plasmablasts enter the bone marrow, where they differentiate into plasma cells, capable of producing antibodies for long periods of time. Memory cells circulate in other regions of the body for subsequent re-exposures to the specific antigen, allowing for rapid secondary response. The exit of lymphocytes from lymph nodes is regulated by S1P, or the sphingosine 1-phosphate exit pathway, which is dependent on the expression of S1PR1 receptor on the lymphocytes. The expression of these receptors on the native lymphocytes are increased if they do not encounter antigens, and they are allowed to leave the lymph nodes. However, if they encounter antigens, their expression is suppressed and they are retained in the organ for further division and differentiation. This is the basis for some immunomodulatory drugs used in the treatment of multiple sclerosis. The thymus is a lymphoepithelial organ located in the superior mediastinum. It is derived from the third and fourth branchial pouches. Common lymphoid progenitor cells destined to become T cells invade the thymus and continue to differentiate until puberty. The thymus then involutes into adipose tissue unless it is re-stimulated in specific conditions. Similar to lymph nodes, the thymus is surrounded by a capsule and is further subdivided into lobules or cortical caps by trabeculae. These are not true lobules but are continuous with the medulla. The cortex is highly basophilic because of the developing T lymphocytes, or the thymocytes, within the epithelial reticular cells, of which there are 6 identified types. Type 1 epithelial reticular cells are located in the boundaries of the cortex with a capsule and the adventitia of the vessels, separating them from connective tissues and serve to isolate developing lymphocytes. Type 2 cells are located within the cortex and are connected to each other via desmosomes, forming stellate cells that express MHC1 and 2 molecules for cell education. Type 3 cells form sheet-like processes with occluding junctions, forming a barrier between the cortex and the medulla. Like type 2 cells, they possess MHCs. Macrophages within the thymic cortex phagocytose T cells that do not meet thymic education requirements. Because the medulla contains larger lymphocytes with more cytoplasm, it appears lighter than the cortex. Type 4 cells form occluding junctions among themselves and with type 3 cells, creating the barrier at the cortico-medullary junction. Type 5 cells are in the medulla and are joined together by desmosomes, forming the medullary framework, similar to how type 2 cells form the cortical framework. Type 6 cells form the thymic or the Hassel's corpuscles. These are masses of concentric cells with flattened nuclei, keratohyalin granules, and lipids. These are functionally active and produce interleukins 4 and 7, responsible for thymic differentiation and T cell education. To separate lymphocytes from antigens, the blood-thymus barrier contains several layers, including a continuous endothelium with occluding junctions, a basal lamina with pericytes, macrophages, and type 1 epithelial reticular cells with their own occluding junctions. This barrier is important to provide a sequestered environment for immature T cells to develop. The process of T cell maturation is otherwise known as thymic cell education and is marked by CD proteins. CD2 and